Our next speaker is Shankar Subramaniam, and in coming up to this talk, Shankar has exchanged a lot of things with me, so I can hardly wait as to what he finally decides to in this presentation. His talk is going to be on bioinformatics and its impact on healthcare. Shankar?
Yeah. Thank you very much. Thanks for the invitation. And this talk is going to be quite different from the previous talks. Here I'm assuming everybody is going to be a patient during various stages in their life. But I'm not going to address the issue of when you're a patient. I'm going to address the issue of your whole body issue based on the genome and how you're going to, have a constant record of your entire life and probably your ancestors as well. So, and so I'll talk about the following issues. I don't know how much time. I'm waiting for a pointer, but bear with me. I'm going to talk about the following issues. I won't have time to address all of these. But I think the fact this morning, your chancellor said that the fact human genome is sequenced has made a huge difference to our perception of what we are and who we are and how we deal with our physiology. And I'm going to talk about the impact of genomics, and this is going to form the first part of my talk. The second part of my talk, which I'm entirely sold on, and this is one of the things that's 20 years from now you're going to absolutely see more and more and more of this, is systems approaches to molecular medicine, as opposed to individually treating one disease or individually collecting one piece of data. You're going to see a whole systems approach. It's a holistic way of looking at medicine. And then the new technologies that are emerging in molecular medicine are going to play an increasing role in terms of how we deal with diagnostic, how we deal with healthcare, how we deal with even developing drugs and so forth. And I'm going to tell you a little bit about the new technologies if I have time. Then, of course, there are whole sets of issues dealing with tracking. And we heard a little bit about this morning in terms of various systems for tracking, RFID-based systems, and LIMS, information management systems. You've got issues of structuring knowledge. We heard already the need for interfaces from Paul, and the need for defining things so that people can understand this in the same way as opposed to in a different way. And then I will skip these things. I won't have time to talk about it. We are going to hear more about imaging later on today. So I will talk about the first three issues at some length. The impact of genomics on human diseases. I want to start with a really interesting example that I came across. Here's a patient. This is from New York Times in 1999. This patient's name is Warren Wegley, and he went to a hospital in Cleveland. And he said, what did I do? I went the wrong direction. And for him, he had an alteration in one of his genes. He had congestive heart failure, and he had alteration only in one single gene, which dealt with one single change. And he said, I've never really gotten ill, and I've always recovered from everything instantly. And this is how we went into the hospital. And they found out that he could not be treated with the standard things you give for congestive heart failure. And in fact, he had this one gene change. They genotyped him and found out that this one single gene change, which is a replacement of one amino acid, isoleucine, threonine in wild type to an isoleucine, if you know amino acids, is a replacement of a single amino acid, a single change in nucleotide. That made him unreceptive to the standard drugs that are being given. And he was told that you need to have a heart transplant. And he had a heart transplant, and he lives even today. And that's mainly because of the fact they could genotype him and find out. This is the extreme case of personalized medicine. So the question is, why is it that, how do we know whether you or I or anyone else has the same kind of problem when we go to a hospital with a particular disease, whether we are susceptible to a drug or whether we are not susceptible to a drug? And so the answer to that lies in our understanding of not the entire genome. We're not going to sequence every human being's genome. You can't sequence 6 billion people's genome, but you're going to find out significant variations in what are called polymorphisms. And the majority of the polymorphisms, we want to assess the variation between individuals. And our major interest lies in largely single nucleotide polymorphisms that cause diseases. And if you look at a map of about, currently today, for example, in February 2001, for example, there is about 1.4 million SNPs that were mapped. And this gives you information specifically on what changes occur in individual genes or proteins in your cells and how they make you more susceptible to diabetes or more susceptible to Alzheimer's or congestive heart failure and so forth. And in fact, when let me skip this slide because majority of you know what SNPs are. I just want to point out a few things. Of these SNPs, 4 million are common SNPs with both alleles of each SNP having frequency of about 20%. 2.4 million SNPs have been discovered in the human genome. in July 2001. In fact, Eric Lander's famous article says that there will be roughly about 1.42 million SNPs that will be identified, and these will give information about individual variation across our genomes. And this is an explosion of SNP data. As you can see, that there's a SNP consortium, there's a HapMap consortium that maps out all this information and documents it. So we will be able to go in and have personalized medicine. We'll have a chip that says, in addition to electronic health records, we'll have a chip that says whatever genotype, extent is. So when it comes to understanding diseases, there are diseases that are monogenic diseases. And in fact, identifying a monogenic disease, single nucleotide polymorphism, is the goal of this research is to find out what are these single monogenic diseases, what are the polymorphisms that can cause a disease. And that will allow us to understand the disease process. For single gene disorders, we have the technology. I went the wrong direction again. So if you go to cystic fibrosis, for example, there's just a single mutation in your chloride ion channel this is your genome. And this is one single change, and that causes cystic fibrosis. So identifying these and knowing who is susceptible is a very important point. This comes into the broad category of genotyping. So how SNPs are used to find genes contributing to diseases? I'm going to give you a few examples. I want to walk you through, since time is short, I'm going to walk you through examples. Here's an example of, there are regions which are coding SNPs. Now there are regions in your amino acids that code for these changes, like in the cystic fibrosis case, or sickle cell anemia. And in those cases, you want to know exactly how this influences what drug will act on your cell, what drug will be efficacious for you, and so forth. Then there are synonymous SNPs. There are things that change that will not make a difference to your change in the structure of the protein or sequence of the protein, but nevertheless will change the content of your genome as a whole. And then there are non-coding SNPs. We don't even understand what this is. In the next years to come, we'll understand what these are, how they regulate gene expression, how they change genes in your cells, and so forth. Here's examples of SNPs that cause diseases. Here's primary hypomagnesemia. Patients unable to maintain sufficient levels of magnesium. It's caused by affected one single gene, sodium potassium ATPase subunit. And mitochondrial SNPs, there are many, many known cases. I'll give you examples of how we generate these SNPs in a minute. And then there are breast cancer associated genes. You know very well about this. Silent mutation in coding exon affects splicing. And so this changes your your breast cancer potential. There are examples of glucose dehydrogenase and favism. This is one example where people, 10% with inactive enzyme experience lysis of red blood cells when consuming fava beans. So you can't eat fava beans. So, so you can know, if you know a priori that these are the causes, these are the genotypic causes, it helps you a lot in terms of understanding what the origin of your diseases are. There is cytochrome P450 mutations, very, very, very important, because cytochrome P450 metabolizes all your drugs and keeps them from becoming toxic. And if you have defects in cytochrome P450, then you're not able to metabolize your drug becomes a lethal toxic reagent. Now, if knowing what kinds of P450s are active in your system, what is specific to you or to a subpopulation of people, allows you to pre-screen drugs and tell you these drugs are okay for you, these drugs are not okay for you. So understanding polymorphisms is fundamentally important. There is extremely little doubt, I'm sure all my colleagues here would agree with me, 50 years from now, every one of us will have been genotyped at various levels, various degrees of detail. How do you do this assays for genotyping? This is a technology. This is a whole slew of technologies available for genotyping. Today, for example, if I want to get genotyped, they will take my genes. There is machines manufactured by Sequinome, which will do a large-scale mass spectrometry of identified regions. It's like fingerprinting. They will take this region and genotype you and say that this is your step, this is specific to you, and this is specific to the subpopulation, and so forth. So this is becoming really a common thing. To give you an idea about how complicated this type of data is, I'm going to give you an example. This is how the data comes out in the instrument. This has got to be parsed into information. So what bioinformatics does is, one of the things it does is to develop technologies and methods which will take this kind of raw data and process it into meaningful, useful information. And this is, much, much more complicated and much more data intensive compared to the patient health records that you saw. And so how do you deal with this? How do you deal with the ontologies that are associated with it? How do you deal with interpretation of data? How do you deal with statistics of this? is what bioinformatics tries to do. It's still, of course, not reached patients, but it is early in that stage. Today, if you want to find out, if you have a family history of a particular disease, you want to find out what types of polymorphisms are likely to occur in your system, you can go to the genome browsers that have SNP tracks. You can ask the question, my parents, my grandparents, my great-grandparents suffered from, some form of stroke when they were 50 years old or 60 years old. What types of polymorphisms exist? You have the ability to go and browse and identify it, ask your physician, and you go to hospital to genotype you for looking for that loci, and ask the interesting question about whether or not you have that or not. So this is really becoming a large database structure. In fact, there is a number of cases. CGAP is another place where you can find genetic annotation initiatives for polymorphisms. You can go to Cancer Genome Anatomy Project and find out what types of cancer you're likely to have, what types of propensity you have for a given cancer. You can monitor the changes that happen during this cancer. I'll show you later on today. And then you have different types of SNPs. You can look at, for example, individual genes, Down syndrome, critical region gene, for example. You can find what are the SNPs, whether you have it, and so forth. So medicine, the genome has allowed you now to catalog and identify parts lists that vary within individuals. And this is really a profound thing since the time that you've come across. Anyway, this is the SNP Consortium to give you an idea about the websites and so forth. SNP Consortium gives you information about 1.4 million SNPs. And this is the HapMap project, the haplotype project. I'm giving you information on how people store these kinds of information. Okay, I should go on to tell you about my, how does bioinformatics play a role in identifying these things? You think that this is all very straightforward. It is far from being straightforward. So, for instance, mitochondria, which is the storehouse of energy in your cells, it also is a storehouse of diseases. There are lots and lots of diseases that come about. I'll give you examples of technology and methods that we developed, in fact, in my lab, for instance, which allows you to look at genes which are identified in specific diseases. I'm going to go through it a little bit rapidly here. So this is, for example, a pipeline of calculations you can do from the genome to identify what are the genes and what diseases do they encode. I'm going to give you two examples. For instance, what we did was we took the entire proteome of mitochondria, which is all the proteins that are expressed in the nucleus, but that's localized in the mitochondria, and we identified their roles, functional roles. Once we identify the functional roles, you can start asking questions about what are the different disease possibilities that exist. I'll show you two examples of this in a minute. So we took and looked at this process called lysine degradation. Don't worry about the very details of the science. It's essentially a process that happens in your body every minute. And so this is the amino acid degradation of lysine. We identified 4 novel genes, and these 4 novel genes by comparative genomics, by comparing human with mouse and rat and other animals. And these 4 genes are involved in serious diseases. In fact, known in human diseases, hyperinsulinemia, hyperlysinemia, for example, happens because of this particular gene. And we start using the power of genomics, using comparative genomics to identify these genes. Now we can ask questions about polymorphisms in these genes to see if there are polymorphisms in these specific genes that can cause diseases or that can prevent cause of diseases. In fact, which population is not susceptible to this disease. So we can start exploring these kinds of questions. Another example of this disease is Brenner syndrome. We identified 2 genes involved, one in Brenner syndrome and one in Sjögren-Larsen syndrome. And these are in mitochondria, and they're involved in threonine degradation. Similarly, we're involved in methionine degradation. So I can go on and tell you about this thing called maple syrup urine disease. So you eat maple syrup and you have a urine disease associated with that. So now the question is, not all of us will have it, but some people might have it. So you can start identifying your diseases using these kinds of genome-based approaches. So I'm going to fast forward here for a bit and tell you about the technology. What types of technology do we have to identify these? So we built an array-based technology. So essentially we want to know, does your genome, does your cell have a given enzyme or not? So we can look at a whole metabolic pathway. We can take, even if we didn't sequence your genome, we can take a metabolic pathway. We can coat these wells with specific motifs that correspond to these genes. And then ask the question, take lysate from your cells and ask the question whether or not your cells contain this enzyme. So we can use this, an example of this, for example, we looked at we used this chip called a nanogen chip for doing this. and we looked at lactose metabolism in Vibrio cholerae, for instance, and we can identify this genes exist or a given pathway exists in your body. So we can start probing your cells, not just for genes, but for the function of these genes across a pathway. So this gives you the ability to ask incisive questions. My next example will illustrate this even more dramatically. So I want to move on to tell you that not all diseases are monogenic diseases. So we have majority of the diseases we are going to come across are multigenic diseases. So we are going to, it's a combination of the genes and environment. For example, stroke, breast cancer, diabetes, everything is coupled. I mean, my colleagues know a lot more about this than I do. Everything is connected to each other. How do we use a genomic perspective to understand this? When you go to a hospital today, a physician today, you complain of a cold or a sore throat, they give you a given medication. When you complain of, for example, if you have diabetes, it causes all kinds of other syndromes associated with that. Now, how do you monitor, how do you deal with this? How do you deal with asking the question why a given drug doesn't work for you specifically? So these are the questions that we try to answer using a systems approach. In fact, we think of the whole thing as a cell functioning as a unit, a tissue functioning as a unit, your organ functioning as a unit, as opposed to individual genes and proteins and drugs and so forth. So I want to walk you through two examples from my lab. And let me skip that. The first one I want to deal with is I want to deal with disease models, give you a systems biology perspective on diabetes. My colleague, Jerry Olewski, who is a physician, he's a clinician, he works with diabetes. He's got phenomenal amount of data from patients. He's collected all this wonderful data from patients, anonymized, of course. And so now you ask the question, how can we use this patient samples, tissue samples, and data in order to make sense out of the systems approach towards understanding diabetes? I'm going to give you an example of this. For example, diabetes is, all of you know, the 6 to 7% of U.S. population suffers from diabetes. 800,000 new cases every year. It's a leading cause of adult blindness. It's a leading cause of renal failure. It's a leading cause of amputations and increased CVD incidence. So all of this comes from one syndrome, I mean, or at least this is a complex syndrome. In fact, the root cause of all of this is insulin resistance, and insulin resistance is connected to every one of these types of disorders. So we're going one step even further. Now we're saying this insulin resistance causes not just diabetes and related diseases, but diseases, all other kinds of diseases. Now, the core central thing, today if you go to a physician with diabetes, they're going to give you one of 3 drugs. Actually, one of them is now banned, so therefore one of 2 drugs. They're called TZD drugs. And they all act on this receptor called PPR gamma receptor. What this does is it affects 3 different tissues. It affects adipose tissue, liver tissue, and muscle tissue. In fact, if you treat PPR gamma agonists or drugs, it increases glucose uptake and lipid uptake and storage, energy expenditure, and so forth. And they're all talking to each other all the time. In fact, if you look at the network, there's metabolic pathway, there is signaling pathway, there is gene regulatory network. All of them talk to each other. And this crosstalk between this causes all kinds of interesting problems. So when you treat them with TZDs, then the TZD treatment essentially changes expression in adipose, changes metabolism in the muscle, and changes in the liver, various types of processes in the liver in terms of lipid uptake and so forth. And so this is kind of a systemic approach towards dealing with this. So the discovery of TZDs happened entirely accidentally. People then say that I want to understand what is the connection between these different phenotypic networks or pathways. And so they tested these different drugs, and TZDs came out to be good drugs. Now, in fact, we know this for a fact that there are already molecules for which drugs are being developed. Leptin, for example, TNF-alpha, for example, interleukin-6. We know this very well. But the question is, how do we understand this at a systems level? The question I'm posing is very simple. The question I'm posing is, if you not one gene, but if you've got a connection of all these things involved, what is the best treatment that can be offered? Okay? And what is the treatment that can be offered to a subpopulation? So here's an example of what we do. We compare data from humans, rats, mouse, knockouts with different drug treatments, different time periods, 3 months, 3 weeks, 24 hours, and then with insulin treatment. And we want to ask the question, what happens in the cell? Okay, the technology is there today to do this. What the technology lets you do is to look at changes in gene expression. We can take a microarray chip. I'm sure many of you have seen a microarray chip. We can take 25,000 genes or 30,000 gene probes on a chip. We can start asking which genes are differentially regulated in these cases, and using that, we go back to biology and build the network. Okay, that's the gestalt of what we do. And so if you look at these genes, immediately we find out that fatty acid metabolism is affected, cholesterol metabolism is affected, not surprising, fatty acid cholesterol transport is affected, and we can look at all these comparative analysis to look at this. We can look at pathways, we can look at how triglycerol is getting metabolized. We can identify absolutely key genes that are differentially regulated in controlling triglyceride treatment. In fact, rosiglitazone, which is a treatment that's offered, upregulates transcription of every enzyme involved in fatty acid oxidation. This is how you're cured, you're kind of treated for diabetes. Increased fatty acid oxidation improves lipid profile, which is why you get your lipid levels down. And results suggest that transcriptional control is important, and this is how the cell maintains its metabolism upon your treatment. But not just we know that how it does it, we know precisely how it does it. Allows us to go in and look at specific drug molecules. In fact, we developed an entire new strategy for doing analysis of this. I won't have time to talk about the mathematical analysis of this, but I'll tell you about, we have data from 60+ patients on 4 quad muscle biopsies, pre-clamp with insulin, post-clamp with insulin, treated with 3 different drug molecules, and we did analysis of extensive things, very interesting results, and I'm going to show you just a summary of the results. We compared between obese and normal, treated and untreated. So there's A versus B, A versus C, B versus D, and C versus D, lean, obese, diabetic, and non-diabetic between patient groups. And then we looked at specific effects of insulin on these enzymes and the signaling pathway, and we find really interesting targets for drug discovery and pharmacogenomics. And in fact, this is a complex central carbon metabolism. We can look at the central player, key player, for pyruvate dehydrogenase kinase, PDK, which is now a major drug target among major pharmaceutical industries. So starting with, in fact, a treatment that people accepted, we can start now deciphering what are the other alternate treatments and pathways that we can target at. Non-diabetics versus diabetics, in fact, affect non-diabetics, downregulation of this enzyme, which causes their failure in your metabolism. This is exactly what happens. 2 minutes more, okay, I'm going to go fast. So if you're diabetics, you clamp, you get downregulation of PDK4. And so you can compare these kinds of effects between patients. Physiologically, you can explain what is a sensor of intercellular glucose, how is it changing, and you can start having devices that will monitor specific enzymes. Here's a complex network. I'm going to skip this part and show you one more thing. This is very important, and I want to show you this. Today, in fact, we can do gene therapy for these kinds of things, at least in mice. So we can say this gene is being downregulated, you want to upregulate it. Here's an example of lentivirus-mediated gene transfer in these adipocyte cells, the fat cells from mouse. And how do we know it works? We know it works because we can tag along with it a green stuff, and this green stuff shows up here. Okay? So this is not a fiction. This is not George Orwell's 2004 Space Odyssey, but this is reality. And in fact, we showed using this lentiviral technique in mouse, we injected this in a diabetic mouse. We injected GLUT4, and we can see the consequence of fold increase in insulin right away. Compare this with this. And so you can start doing treatments just targeted specifically to cells, targeted specifically to genes. Okay? And I'll summarize in the last slide. Disease models. I'm going to skip this. This is infectious diseases we can treat and work in collaboration with Christopher Glass. We have started identifying specific pathways involved in salmonella, in gram-negative bacterial infection. We started looking at networks involved in these things. We identified a large number of these players. We can look at, in fact, I'm going to show you these pathways. I don't know if I have time to show you these pathways. Then I'll skip over to stopping at this point. All right, I decided to stop because there's too many things going on.
I just want to say that all the presentations will be on our website a week from now. So I know that there's much more material in these presentations from all of our speakers. So don't fear, you'll be able to see it at some point in the future.
Yeah, thank you. I covered 30 of my 146 slides. So
Well, there are 2 things I want to say. When you said go faster, it scared me. And two, I was glad that Brendan's here because at least one person fully understood what you said. So very, very interesting presentation. Question, first one here.
Yeah, well, without a microphone.
May I have a microphone?
It's pleasing all these things in this nice presentation.
Oh, I'm sorry. I'm sorry. I've screwed up. I want to go to the panel first. I got lost. I'm sorry. I saw your hand and I responded. I'm sorry. Maybe we'll start with Paul.
Very talk, of course. You talk about gene therapy and picking out the SNPs. How are we as good at picking out the potential complications in messing around with nature's pathways? I know the disease messed it up, but does our unmessing it have predictable results? So I draw back on gout is caused by an excessive inflammatory reaction. And so colchicine basically cuts the white cells off at the knees so they don't get there. But who would have dreamed that you'd actually put that in humans and it doesn't have that much side effect? But how do we predict the consequences and complications of therapy?
Paul, this is a great question. So, in fact, Paul is, providing us the greatest argument for a systems biology approach as opposed to an individual gene approach. So it could be an individual gene, but you do not target an individual gene. You want to find in a network, you want to find the point that's got the most influence but least connected. Okay? And you want to test it in animal models, and even then it may not work in humans, but you can test it in tissues. So the technology, I didn't get to my end of my talk, technology today allows you to not do mouse and chimpanzee and other testing, but allows you to do human tissue testing. I'm a great proponent of stem cell research for that, and because it allows you to really do this exercise of testing in a real tissue culture, in a real environment. Yes, it's not the entire physiology, but nevertheless, it is a significant part of the physiology. And so therefore, you could do this testing. So the answer is yes, there are side effects. This is research. This is far from being clinical medicine at this point. We are not anywhere ready except in very, very few cases to take it to a phase 3 trial. Nevertheless, this is where the future is going because you want to ask the question, how do I decipher the whole network involved in normal versus disease function? There's another issue which I didn't refer to, which is that there's nothing like normal versus disease. It's a continuous progression of things. And so you want to ask the question, at what point in your move over of a Gaussian distribution from normal to another Gaussian distribution for disease, where are you at that point? And so this is where networks allow you to start investigating specific genes and enzymes and gene products. I hope I answered your question.
So if we believe that over the course of the years ahead that increasingly the individual patient's data will include genomic and proteomic information, to what degree do we understand how systems such as Paul presented will be different? I mean, what will be different about them 5 years out, the classical electronic health record?
Yeah, an excellent question. So, what Paul presents today is terminology and language and linguistics that most patients understand. Now, the moment I start talking about crosstalk between pyruvate dehydrogenase kinase and some other protein or enzyme, the patient is lost. They don't have the biology in order to understand it. So this is fundamentally a big different issue. So structuring this knowledge, making it, translating it from a scientist like me to a physician who understands this stuff to a patient who understands even less is really a significant challenge. So this involves structuring of knowledge. This involves our being able to decode, go from genotype to phenotype to disease in a seamless way, which requires a large amount of work in terms of our communication capabilities between scientists and physicians and patients. And so this is not going to happen overnight. It's going to take a long time to happen. And even then, we'll only get a nutshell synoptic information. We won't get information that really is in-depth.
I enjoyed your talk very much also. Just a comment and then a question. As far as comment, recently there's been a big push towards trying to have a patient identifier or a master patient identifier in the U.S. And as part of the HIPAA legislation, that was introduced and then that failed. And so it'll be fascinating to see in these next 50 years or 25 years or whatever how we move from a culture in which we can't even have a patient identifier number to one in which we actually have their entire genome that's sequenced and stored someplace. And so it'll be interesting. The question that I had was how can we change our electronic medical record so that in the future, as we have this information available to us, it's not patient-centric, and so that we can actually do meaningful studies where we compare phenotypic information on patients, radiology results, lab results, and have some way across the entire system of being able to take advantage of the database and actually mine this incredible, wonderful database that we could have. Our electronic medical records today aren't going to allow us to do that.
Excellent question. I didn't get to the very last part of my talk. The last 20% of my talk dealt with how do you structure these complex things, how do you create databases. In fact, we created a database of what are called molecule pages. It's essentially every type of information you can think about a single molecule in your physiology. And this contains about 420 Oracle tables. And it's a very complex database. It required about 15 people to develop. It required programmers. It required Java programmers. It required database programmers. It required interface programmers. So this is not a trivial task. It's very complex. But we have to address the bull by the horn. So therefore, we don't have a choice. So this is where, for example, your healthcare system here, your department here is involved in terms of structuring these things. So we have to go from, it's a slow process. I don't have solutions, magic bullet solutions, but it's a long way in coming.
Great talk. The good thing is I got to take some of my genetic slides out of my talk for later, too. I have one main question for you, and that is, it sounds like, one of the challenges, again, with radiology, if you graduated 20 years ago in radiology, you didn't have the advantage of learning MR, and then you have to go back and train yourself all over again. How on earth, as these things go from bench to lab, are clinicians going to have to figure out which of the 1.4 million SNP alleles to be taking a look at? And obviously I'm leading into it, does this need an expert system? If we don't have an expert system, will it not take off in clinical practice? Or what are your thoughts along that way?
And this is, again, a very good question. In fact, I think John's colleague will be able to answer this question a lot better because they have a funded grant on bench to bedside on dealing exactly with that. Obviously, a physician is not going to look at 1.4 million SNPs. What if when you're genotyped, and nobody is going to genotype you and say that this gene has this change, that gene has that change. You're going to have, just like you're having markers for things, you're going to have markers. You're going to have lots of markers. We say, for example, when a fetus is born, for example, today we have markers for about 700 plus potential diseases, childhood diseases that a fetus can have. So you'll have markers of this kind based on not a protein marker, but now based on the gene SNP. And this will be tagged on so that when you get a specific disease, it'll be not an expert system, but I would like to call it as a knowledge system, which would tag on. For example, you imagine you get an onset of cancer, it'll tag on and go and identify those specific genes which have polymorphisms and say that this individual has the following, so therefore you have to do this following treatment. It's just like the case of Warren, the patient I showed you from the New York Times article. They could zone in because the drug molecules are for adrenergic receptors, they're catecholamine type receptors, drug molecules, so they could go and look for polymorphism in specifically that gene and say that this patient has a mutation. Okay, so it will be specific to each given disease, and there will be markers. Already it's happening, so this is not something that's 50 years from now. It's already happening in large number of cases.
Any other questions or comments from the panel? If not, okay, Pat.
Well, maybe the 50-year thing, I'm going to be checking out what you're talking about. So how much sooner than 50 years will we progress from genomics to proteomics to phenotype to diagnosis and treatment?
I did not mean to imply that we're going to have to wait exactly 365 plus 1 day, 4 times, 50 years. I didn't mean to imply that. What I really meant was in 50 years it'll become a lot more routine and automatic. Even today, genetic counseling is very common, as you know. Even today, for example, they do assess various proteins in order to assess your disease. Diabetes is a very good example. There are a number of markers which are being assessed constantly, other diseases too. So from going from genes to proteins to phenotypes, the problem is the systems approaches have not taken control yet. And so what I meant really was that in 50 years' time, you'll see a lot more application of systems approaches, but it's happening already.
Okay, question here.
It seems that the human body has a better internal EPR than the hospital for the moment. It's working better. So the conclusion is that your genetic work will not be possible without huge efforts in IT. That's number one. So I will put three questions. The first is, how can we merge this bio-IT with the hospital IT and the clinical IT of today? Second question is, how does it affect these medical specialties, and how should we derive there rules and criteria useful for the current clinical work today, for evidence-based medicine, clinical pathways, and so on? Because I don't have any vision how this can work today. And the third is, we have seen that security and privacy of the patient, well, seems that patient record is a small peanut compared to what information and personal privacy is inside the genomics. How can we handle this?
They're all very good questions. All three questions that you asked are very good questions. Let me track them in reverse, mainly to place the emphasis on the broad scope. So by knowing SNPs today, we don't target an individual patient. At this point, you can say a subpopulation of people who have a deficiency in this beta-adrenergic receptor will not be susceptible to this catecholamine agonist. Okay, so you've got broad categories of people, subclasses of people. Now, you may fall into the category, or I may fall into the category, and that's the next level of difficulty that we have to deal with. The issue with reference to how do you tie in genome-driven informatics to clinical informatics is a huge nightmare. It's not trivial. Where it starts today where it's done is in genetic counseling. For example, you have somebody who's got an albino syndrome, for example. Then you have genetic counseling. You talk about how, you start explaining about the consequences of that, the impact on healthcare down the road and so forth. So there is already a bridge, but you need a counselor. At this point, it is not automated. It is not something which I can go seamlessly across. Take diabetes as an example. Suppose I'm a patient, a diabetic patient. I would like to not only monitor my insulin levels and write them down in a notebook or an electronic health record, but I also would like to measure my GLUT4 levels. I would like to measure my other types of leptin levels and so forth. And this can be done today. It can be entered today, and it can be correlated statistically at this point. We don't have a first principle understanding. You can do statistics to correlate between the levels of these and other types of physiological consequences, phenotypes, if you want, clinical phenotypes. I can correlate them. And I can say when I see higher levels of GLUT4, I see the following things correlated. Today you can do that. And there are a lot of systems which start doing that already. Even from radiology point of view, there's lots of systems that start doing that. But it is not far from clear at this point how to integrate this to informatics, and a large number of efforts are focused on how to, in fact, NIH is constantly asking us, how do we relate, bioinformatics and clinical informatics, and this is one of the issues why I think I was invited to talk about these issues. It's far from being trivial. Now, your second question in terms of the privacy of this information, this is a fundamental problem, especially in the United States, because we have a system where the health maintenance organization will immediately cut you off if you have something, and then so you need to really constantly worry about this. HIPAA, for example, allows you to regulate some of these things, but nevertheless, this is an issue. So scrambling and having access control to medical records is not a problem. That's very easy. But what is not easy is who gets access to the access control records. That's the issue that you need to worry about. And this is why I asked the question to Paul about this. This is a fundamental issue. And I don't think scientists have a solution to this. This has got to come from the society as a whole.
My question refers to the SNPs in the regulatory region of the DNA. First of all, the manifestation, of course, is not as obvious as in case of the structural gene region, because the changes are, of course, differential rather than absolute.
Correct.
Now, my first question is, are there any diseases associated with it? And second, how would you study that using a systems approach is perhaps the only way, as far as I can tell.
Very, very good question. So what is being referred to is that you have your genome is 3 billion base pairs, give or take a few million. And, and so out of those, there are about maybe 30,000 coding genes or so. I mean, with lots of splice variants, of course, but there are 30,000 odd coding genes. Now, your polymorphism may occur in the coding region, or it can occur outside the coding region that regulates the coding region. And how do you know if it regulates? Are there diseases? Yes, there are many examples of diseases where the regulatory regions have the SNP and that regulate change in expression of a given gene product and so forth. How do we know? How do we deal with this? The only way to deal with it is what you pointed out, which is to think about it as a systems approach, because if you have a regulatory region, it's not controlling just one gene, it's going to control probably hundreds of genes. You want to ask what are the other 99 genes doing, and how are they regulated, how are they controlled? So this is why systems approach is absolutely the only way to go. And gene expression chips and what are called chip-chip methods. I mean, again, I had a slide on the chip-chip method, which is chromatin immunoprecipitation DNA chip methods. These methods are now starting to ask the question, who talks to whom in the regulatory pathway? Who controls whom? And who discusses with whom in terms of making a gene express? And so if you know this map, then we can start answering the questions that you said in terms of networks and saying that this SNP in this regulatory region is going to cause the following changes. So we are ways off from it. We can do it in yeast. Not in human yet.
Questions on this side? Back there? Answer.
A great talk. I can understand the issue with respect to structuring the knowledge. It's actually a very common generic problem. But how to cope with the evolving of the knowledge?
How to cope with what, sorry?
With the evolution, the evolvability of the knowledge.
Yeah. Very, very good question. So there are two aspects to the evolution of knowledge. One aspect of the evolution is that where your structured vocabulary and ontology still is valid for the evolved knowledge, in which case it is trivial, it is not a very difficult thing to do. However, if your entire semantics of dealing with things get changed, then how do you deal with it? In fact, if many of you may know this, there is this famous project called the Semantic Web Project, which really tries to, you're involved in that. And so the Semantic Web Project essentially tries to associate meaning and concept or associations with reference to websites and so forth. So we, in fact, are working on, in collaboration with the computer scientists, are working on creating a semantic ontology approach where you build a graph and then you imagine you are an end user and you find this concept has changed. You can interactively modify the graph so it then reflects in your database. Now, there are fundamental issues, computer science issues with reference to this. Sometimes the ontology will affect the table structure. Sometimes it will not affect. If you're a relational database, your normalization goes out of the window. And so there are all these kinds of complex issues. But I think we can deal with this. Computer science has to deal with these issues. They have gone away. Computer scientists are starting to realize that biology is a domain because there's no longer apples and oranges tables. It's now gone into very complex, convoluted relationships. And so this is where I think computer science research has to focus in terms of looking at interactively evolving ontologies and their impact on data structures in databases.
Questions or comments on this side? Dave?
Not to harp on the privacy thing, but has anyone thought about, in the same way that we have used body parts in terms of tissue organ donations, setting up systems where you could donate your genome and your medical history for the purposes after you're gone, so that you'd have a repository of people who are interested in doing that?
I mean, I don't think there are projects of this kind. There are projects like freezing your own sperm and things like that. And the sperm, of course, contains all the information about your genome. So, but on the other hand, I don't think there are targeted projects, but maybe Healthcare Canada can come up with a project of posterity genome project. You can call it as the PGP.
I'm worried about you, David. Up here.
Phenotype is a result of the genotype interacting with the environment. Why? Then I'm wondering why we don't spend as much efforts in typing the environment, because it could be that it is as much influencing our ultimate health than the genotype. We more and more see that even identical twins differ very fundamentally.
You asked this is a very, very interesting question. So this is always the nature versus nurture debate framed in a slightly modern context. And so here's the response to this in two types of responses. Let's take the extreme case where your environment is so dangerous to you that it kills the species or organism, in which case we can do nothing. Genomics can do nothing about it. The common belief based on experimental evidence is that your cells, your tissues, your organs function under certain given restraints or constraints upon them. If you change them dramatically, then they just simply quit and they die. So there's only a finite number of such functional capabilities of the system. So the goal is, I mean, you're indirectly typing the environment into the system through genotyping and phenotyping the gamut of activities that you can normally observe. And then you're not worried about outliers, because outliers are going to kill you anyway. So you're worried about the mean of these things. And so you say the cells are going to function under the common constraint type of things. So we want to indirectly look at how does environment influence, how does, for example, change in glucose, how does change in media influence your muscle tissue. So that we can do today. What we cannot do is to take into account the tsunamis of this world.
Over the side? Questions or comments? Anyone? Okay, on the side? Anyone? Murray. Murray William.
I don't know anything about this area, as you know, but and this may be a very naive question, but looking at some of the things from a layperson's point of view. Now, so far we've heard a lot of information that could address the area of curative medicine. But if one is looking in the area of preventative medicine, would this not be a hook that, say, the U.S. society at large could appreciate, so that this privatization and this concern of personal information not be overcome politically, because then it addresses the general good, right? As I said, it's probably a naive question, but from a layperson.
No, I think it's a great question. I mean, I don't know many of you are familiar with this, something called the Iceland Genomics Project. What the Iceland Genomic Project does is that they have got, Eric the Red gave rise to every Icelandic person in the world. So they have family history starting from Eric the Red. They have got exhaustive family history information. They have now tissue samples from all siblings and relatives and nuclear families and so forth. And they have all the diseases. So they have got the genotype and phenotype, and they have started collecting information on what, in fact, this company called Decode, what they do is they start collecting information on how to relate the genotypes to family history to phenotypes. And so that's not only resulted in benefit for single nuclear families, but it's resulted in benefit overall for society. In terms of, for example, we know specific markers for breast cancer, for male testis cancer, for example. There are specific genes and markers found through this method of looking at the Iceland Genomics Project. So there is tremendous benefit to be accrued by looking at the genome as a whole, as an average, as in a given restrained environment, as opposed to looking at patient records and securities for individual persons. So ultimately, it will come to that. But at this point in time, we don't know enough to say that we know, all the other information, so we might as well start looking at the generic SNPs and generic phenotyping information. This is what the HapMap and the SNP project try to do.
Coming back to consequences for informatics, not every doctor, not even every hospital has a supercomputer today to do these things. Is there any solution in the short run how we can solve this? I see there appearing open sources, grids, other types of databases coming. How should we tackle this problem, or should we wait simply till the teraflop machines are available in the shops?
Yet another very, very good question. So in fact, I was expecting John to address some of these issues as well. And so there are two dichotomous issues that you have to deal with. Retrieving information, for example, is not a supercomputing operation. But filtering information is a large computing effort. I mean, imagine I have a large amount of data in memory. I want to compare your genome and his genome and my genome. That's not doable. That's a large memory-intensive computing operation. So the question is, how do we parcel information into packages that are self-contained yet coupled to other things, so without having latency issues and I/O issues and so forth? It's a complex, very difficult computer science issue. The second issue coupled to that is the communication network. Imagine I'm in a village in Minnesota, and I'm interested in accessing NIH health records as compared to something else, and I have only a 900 baud modem sitting in my house, and I don't think they any longer make 900 baud modems, but let's say hypothetically. So, I mean, and then I need to get this information. I can't download my radiological, I cannot download my X-ray, I cannot download my images or anything like that. I can't do comparisons. So this is a communication networking problem. In fact, we had a symposium in NIH where some of us tried to talk to them about not just investing in terms of the software and the informatics, health informatics system, but also investing in terms of looking at communication lines and so forth. I think it's improving dramatically, but it'll take a while before it becomes a large-scale effort. So the answer to your question is, it requires investment of resources. It requires development of innovative methods for parceling information, yet being able to access all of them. So this is not a very untypical problem in computer science where people deal with this. I mean, if you have large tapes and things like that, and you want to access a specific piece of information in that stored tape, then how to do it. Today, in fact, there are ways of doing this in much more intelligent ways. And so this is where I think computer science comes into picture.
Any final questions on that side? Over there, Dennis? Get a microphone.
It's another layperson question, but I'm sort of reacting to this 50 years that you talk about, because I'm assuming that right now, today, I could find out my own, or any individual could find out their own sort of genetic makeup. And when you add that to the internet today and the information that's available, even some of the research you talk about, isn't that really going to push this thing much more quickly? You know, when individuals start having information that, and then they're going to start seeking solutions out for their particular problem that they may have identified.
So if the first part of your statement is true, then the second part of your statement is fait accompli. But the first part of your statement is not true yet. For example, I cannot say, as a John Doe citizen, and walk into a health hospital in San Diego and tell them, genotype me today and give me tomorrow all the polymorphisms that I have. That's far from being trivial. If it happens, absolutely, you can put it up on the web and you can do all kinds of interesting things. But it's still a technology which is neither there, it's expensive, it is complicated, you've got to provide a rationale for doing it. And of course, if you're Bill Gates, you might be able to do it, but a lot of us don't have that luxury. So nevertheless, if that's a very good point, if you do genotype your information, and then posting it even in anonymous ways would be very useful, because it will help the community start building resources and databases that can talk to each other. So,
how far are we away from a cost-effective way of getting around?
The technology is constantly improving, but we are still ways away from there. I would say it will take a decade before we routinely go in and, I mean, let me give you an example, because the examples are the best way to look at how nobody can look at a crystal glass and say this is 4 years or 5 years from now. But I can tell you for fetus phenotyping, for example, I mean, till about 5-6 years ago, or maybe 7 years ago, you could look at about 20 or 30 different possible diseases that a fetus can have. Down syndrome, for example, you could look at that, and you could decide a priority on genetic counseling at various levels and so forth. Today, you can look at 700 plus diseases, and I'm not joking. This is actually a fact. You can take a sample serum from a fetus, and you can look at 700. So this is dramatically changed and improved. So as technology becomes more and more common, especially when we have finished SNP mapping 1.4 million SNPs and identified, let's say, 20,000 key mutations that are not specific to you, but to a subpopulation of people, then we can start doing genotyping of individuals at that point. So I think we are still far from there.
Question over here. Okay.
This is, again, a lay perspective, and specifically, at least a parent whose son died of cystic fibrosis, and having been very disappointed with the genetic progress, and at the same time disappointed that nutritional and biochemical approaches have been grossly discouraged. From Dr. Douglas Crozier, who was the original definers of CF and then working at the Hospital for Sick Children in Toronto, being discouraged to look into essential fatty acids, particularly the omega-6 group. People wouldn't even do, at the University of Toronto labs, wouldn't do his work. He got kicked out as director. Then, more recent years, DHA mouse, the cystic fibrosis mouse, and DHA, in effect, minimizing the symptoms and that quickly disappearing. Now, if this has any validity, it suggests that
biochemical imbalances in the body potentially could be corrected by certain biochemical intake, DHA and so on. It relates to Roger Williams' idea that we're all biologically unique, and in certain extremes, disease results, if you like. And side effects were being referred to before, particularly with respect to altering genes through viruses and so on. We know drugs have all sorts of side effects. We really ideally want an approach which has minimum side effects, corrects the problem. And to me, this gets to the issue, too, of professional ethics, okay? And that you don't want to be a cooperant in a system which is unnecessarily risky.
I mean, I would like to summarize my talk by saying just two sentences, and which answers your question. The approach using knowledge of the genome is not meant to replace any other approaches for disease that are effective or productive. The intention is not, the idea of science is not doing that. What the power of the genome offers you is exactly what you said, which is to identify or target that process in your physiology which does not affect which does not affect other processes or influence other processes. And so this is the whole goal of systems approaches and network-based approaches. It's not to say that I want to try this drug or that drug which will do the following, which we don't know what it happens. I mean, you look at Vioxx, look at Celebrex. We targeted them towards specific COX inhibitors. I mean, they're COX inhibitors and they're COX gene product inhibitors. So what we are saying is, if we had understood the pathway by which COX operates, it would give us a potential scope for us to investigate this much more in detail, tell which patients, for example, cannot be treated by Vioxx, have a higher heart disease risk, and so forth. So the ability to genotype provides you with additional knowledge about your physiology. It's not meant to replace anything that exists or is beneficial. And this is the viewpoint that 99.99% of geneticists and people who do genomic-driven medicine would take. And they would not say, for example, I just want to abandon something else and do this.
Well, Shankar, thank you. I'm afraid we've run out of time, but Shankar, thank you very much for your presentation. 1 o'clock. 1:15.
Well, thank you for now I understand why it's not. If I could just take a couple minutes of your time. Thank you very much for your attention this morning. The talks are great. I would like to see the speakers, all the speakers, for just a couple minutes, so don't run away on me. And before we break, I'd just like to take the opportunity to thank a few people, especially our volunteers. They were here late yesterday afternoon, feverishly trying to put things together with us. And so we'll acknowledge them first. Weech and Dai.
Please stand up.
Please stand up. Maybe save your applause to the end. There's a few. So Mahesh Shanuda at the back. David Stevens. Bill Malik. And Mike Chen. And we've got some hardworking technical people working here to keep all the presentations recorded, all the mics and everything that's flying around. So Doug Mulholland is part of our crew here at the Computer Systems Group, and hiding away behind the window there, he's waving, is Anthony Robbins. And I'm sorry, I don't know our member from Audiovisual. What's your name? Paul. Paul.
Thank you all.
Now, not in this room. We have another group of people that are working in 1301 for all of the food and the registration. So we have Colleen Richardson, my colleague in crime. She's been here from the start very early this morning and right from the beginning of planning this. And as well, I even dragged in my sister. She's manning the phones upstairs. So I have to thank her as well. And, of course, the AGFA team. We could not have pulled this together without them. Rennie Stacy has been next door. She got all these signs, beautiful signs done for us. Even Yost Aru, where are you sitting now? He even came in last night and was helping out. So thanks to them. And, of course, a very, very special thanks to Helen Chen. We could not have done this without her. She did so much of the planning with us, did so much of the logistics, so that's just been great. And I think that's about it. Right?
Just, we're back here at 1:15. We've gathered back our lost time at the beginning, so we'll start at 1:15 sharp. Looking forward to seeing you again. Lunch is next door.